There is a resin that has been traded across continents for over 5,000 years. It was worth more than gold in ancient trade routes. It was offered to Egyptian pharaohs, burned in Greek temples, carried by the Magi in the Bible, and used in Ayurvedic medicine for at least 3,000 continuous years. You know it as frankincense. You probably associate it with church incense and Christmas nativity scenes. What almost nobody told you is that frankincense is one of the most clinically studied natural anti-inflammatories on earth, and that multiple peer-reviewed trials have found it outperforms ibuprofen for arthritis pain with a fraction of the side effects. The active compound, ACPA, is currently the subject of active pharmaceutical patent races in Europe and the United States. Companies are attempting to synthesize it, modify it, and own it. Until they succeed, you can buy the resin it comes from for $12 a bag. Stay with me until the end of this video. Because the gap between what the clinical evidence shows about Boswellia serrata, the tree that produces frankincense, and what your rheumatologist has probably told you about it, is one of the most striking examples of what happens when a plant cannot generate pharmaceutical revenue. Frankincense, the dried resin of Boswellia trees, primarily Boswellia sacra, Boswellia serrata, and Boswellia carteri, has one of the longest documented medicinal histories of any substance on Earth. Archaeological evidence of frankincense trade routes dates to at least 3000 BC. The resin was found in Egyptian tombs. Ancient Egyptian medical texts describe its use for wound healing, skin conditions, and respiratory problems. The Egyptians also used it in the embalming process. Its antimicrobial properties were recognized empirically long before microbiology existed as a science. The Silk Road carried frankincense from the Arabian Peninsula and the Horn of Africa to India, Persia, Greece, Rome, and eventually Northern Europe. Wherever it went, medicinal documentation followed. In Ayurvedic medicine, the traditional medicine of India, documented continuously for over 3,000 years, the Indian species Boswellia serrata is known as shalaki. It is specifically and extensively documented for joint conditions. Ayurvedic practitioners described it for what we would now call osteoarthritis and rheumatoid arthritis, painful, swollen, stiff joints, with detailed preparation protocols that have been passed down through unbroken clinical tradition. The specificity of the Ayurvedic documentation is remarkable. It is not vague tonic language. It is clinical precision, which joints respond best, how long treatment takes, what dosage is appropriate for acute versus chronic conditions, which preparations work for which constitutional types. 3,000 years of clinical observation, encoded in a medical tradition that Western medicine spent the 20th century categorizing as folklore. In traditional Arabic medicine, documented in Ibn Sina's Canon of Medicine, frankincense was prescribed for tumors, ulcers, vomiting, fever, and as a general tonic for the nervous system. Ibn Sina specifically noted its anti-inflammatory properties for joint and muscular conditions. The Western medical establishment's engagement with frankincense as a medicine effectively ceased when Christianity adopted it primarily as a liturgical substance. By the time European medicine began formalizing in the 17th and 18th centuries, frankincense was incense. The medicinal documentation existed. It was in the Arabic and Ayurvedic texts, but it existed in traditions that European medicine had categorized as other. Modern pharmacological research on Boswellia began in earnest in the 1990s, driven largely by Indian pharmaceutical researchers following Ayurvedic leads. What they found was, by any measure, remarkable. The primary active compounds in Boswellia resin are boswellic acids, a class of pentacyclic triterpene acids. The most pharmacologically potent of these is acetyl-11-keto-beta-boswellic acid, ACPA. ACPA is a specific inhibitor of 5-lipoxygenase, an enzyme central to the production of leukotrienes, which are key mediators of inflammatory response. This is a mechanistically distinct pathway from NSAIDs like ibuprofen, which primarily inhibit COX-1 and COX-2 enzymes. The clinical significance of this distinction is substantial. 5-lipoxygenase inhibition does not cause the gastric irritation and cardiovascular risk associated with COX inhibition. This is why Boswellia can be taken long-term without the gastrointestinal and cardiovascular risks that make chronic NSAID use dangerous. The clinical trial evidence. A 2003 randomized controlled trial published in Phytomedicine compared Boswellia serrata extract to valdecoxib, a COX-2 inhibitor prescription drug for knee osteoarthritis over 6 months. Both treatments produced significant pain reduction and improved knee function. Boswellia showed earlier onset of effect. Valdecoxib was later withdrawn from the market due to cardiovascular and skin reaction risks. Boswellia is still here. A 2008 randomized controlled trial, published in Arthritis Research and Therapy, tested a proprietary Boswellia extract, 5-loxin, against placebo for knee osteoarthritis. The treatment group showed significant improvements in pain scores, physical function, and stiffness within 7 days. At 90 days, synovial fluid analysis showed reduced levels of matrix metalloprotease 3, an enzyme that degrades joint cartilage. Not just pain relief, potential cartilage protection. A 2011 study in the International Journal of Medical Sciences compared Boswellia extract to ibuprofen for knee osteoarthritis. Both groups showed significant improvement. The Boswellia group showed better tolerability. The ibuprofen group showed more adverse events. Rheumatoid arthritis, a 2019 review in the journal Frontiers in Physiology, summarized evidence for Boswellia in autoimmune inflammatory conditions, including rheumatoid arthritis, finding significant anti-inflammatory effects and a favorable safety profile compared to conventional disease. Modifying drugs. Inflammatory bowel disease, a 2001 study published in the European Journal of Medical Research found Boswellia extract comparable to the standard pharmaceutical treatment mesalazine for inducing remission in Crohn's disease, with the Boswellia group showing a trend toward better outcomes. Asthma, a 1998 randomized controlled trial in the European Journal of Medical Research, found 70% of asthma patients on Boswellia extract showed improvement in symptoms and lung function compared to 27% in the placebo group. The breadth of clinical evidence across inflammatory conditions, arthritis, IBD, asthma, reflects the fundamental position of 5-lipoxygenase inhibition in inflammatory pathophysiology. Block this pathway, and you reduce inflammation across multiple organ systems. Let us discuss ACPA and the patent race. ACPA, the primary active boswellic acid, cannot be patented in its natural form. It is a naturally occurring compound. However, synthetic analogs of ACPA, modified versions with slightly altered molecular structures, can be patented. Multiple pharmaceutical and nutraceutical companies have filed patents on ACPA derivatives, modified extraction processes, and specific formulations. Several European companies have developed proprietary Boswellia standardized extracts, products like 5-loxin and aflapin, that are patented not for the compound itself, but for the specific extraction method and standardization process. These proprietary extracts then get licensed to pharmaceutical and supplement companies at significant markups. The resin they are derived from, harvested by hand from Boswellia trees in Somalia, Ethiopia, India, and Oman, costs a fraction of the processed end product. Meanwhile, the clinical evidence that validates these extracts applies equally to high-quality standardized Boswellia supplements available from multiple manufacturers at a fraction of the proprietary product price. The structure of medical education and pharmaceutical promotion ensures that most rheumatologists, the specialists who treat the arthritis conditions for which Boswellia has the strongest evidence, have never reviewed the clinical trials. There are no pharmaceutical sales representatives visiting their offices with Boswellia trial data. There are no conference sponsorships. There is no CME module on boswellic acids. The clinical evidence sits in peer-reviewed journals, including major rheumatology and gastroenterology journals, and is systematically absent from the clinical conversation because there is no commercial infrastructure to push it there. The global anti-inflammatory drug market is projected to reach $120 billion by 2030. It is built on NSAIDs, COX-2 inhibitors, and biologics, all of which carry significant adverse event profiles with long-term use. The resin that a 3,000-year-old medical tradition prescribes specifically for joint disease and that clinical trials have validated against prescription drugs is sold in health food stores for $12 a bag. And stay with me, because I want to tell you something about the supply chain of frankincense that nobody in the supplement industry is telling you. Before practical guidance, the supply chain issue, Boswellia trees in Somalia and Ethiopia are being overtapped to meet growing global demand. Trees are being bled of resin faster than they can recover, and Boswellia populations in key growing regions are declining. When purchasing Boswellia, look for suppliers with transparent sourcing, ideally with sustainability certifications or direct farmer relationships. This matters both ethically and practically. Over-harvested trees produce resin with lower boswellic acid content. For therapeutic use, the critical variable is standardization. The clinical trials that produced significant results used extracts standardized to specific boswellic acid content, typically 65–70% total boswellic acids, with particular attention to ACPA content. Generic products listed simply as Boswellia without standardization information are of unknown potency. What to look for on the label: total boswellic acid percentage, aim for 65% or above. ACPA content specifically, higher ACPA correlates with stronger 5-LOX inhibition. Products like 5-LOXin are standardized to 30% ACPA. Comparable standardized generics are available. Dose: clinical trials typically used 100 mg to 250 mg of standardized extract, 2 to 3 times daily. The effect is not immediate. Most studies show significant results beginning at 4 to 8 weeks of consistent use, with maximum benefit at 90 days. This is not a pain medication in the pharmaceutical sense. It is a modulator of inflammatory pathways that requires time to shift the underlying inflammatory state. For acute joint pain, combining Boswellia with curcumin, another well-studied natural anti-inflammatory with a complementary mechanism, is supported by clinical evidence and is standard in traditional Ayurvedic practice. The combination hits inflammation through both COX and LOX pathways, mirroring what expensive pharmaceutical combination therapies attempt to achieve. The resin itself, raw frankincense tears, can be chewed directly. This is the oldest preparation method. The resin is slightly sticky, with a complex balsamic, slightly citrus, slightly pine flavor. Traditional Ayurvedic and Arabic medicine use the raw resin both internally and as an external application. Modern standardized extracts are more reliable for therapeutic dosing, but the raw resin consumed as food, as it has been for 5,000 years, provides a baseline of boswellic acids as a dietary component. As incense, yes, burning frankincense resin has been shown in multiple studies to reduce airborne bacteria and fungi, and the inhalation of boswellic acid-containing smoke may have anti-inflammatory effects on the respiratory mucosa. The use of frankincense incense in religious and ceremonial contexts across virtually every major ancient civilization may not have been entirely metaphysical. 5,000 years of trade, worth more than gold on 3 continents, prescribed by the physicians of Egypt, Greece, Rome, and Ayurvedic India for the same conditions. Multiple peer-reviewed clinical trials showing it outperforms prescription drugs that were subsequently withdrawn from the market for killing people. $12 a bag, growing in trees in the Horn of Africa and the forests of India. And your rheumatologist has probably never mentioned it. The incense was always medicine. They just didn't want you to notice. If this matters to you, subscribe. Every week, the resins, the barks, the roots that 5,000 years of human medicine knew by name, and that the modern system filed under not profitable enough to mention. Subscribe. I'll see you in the next one.